Hi, welcome to the Entrepreneur Spotlight hosted by the Pennsylvania Biotechnology Center. I'm Lou Casa. I'm the Executive Vice President and Chief Operating Officer of the center. This program is one of my favorite to host as we spotlight companies in our community and help them make vital connections in the industry, which could be anything from investment to consultation. It's our version of the Shark Tank, and we have some great sharks lined up this year. Before we start, we have some housekeeping items for everyone. The housekeeping items actually got moved to another piece of paper. There we go. Having all kinds of technical difficulties here. But anyway, to submit questions, click on the live Q&A button on the right-hand side of your screen. To view the program book or any information about today's presenters, click on the session information button right above the live Q&A button. And finally, to view today's program in full screen mode, please click on the diagonal arrows icon in the upper right-hand side of the presentation window. We've prepared a video about the PABC, and before we start it, I want to mention that our model is very unique, and it's what we like to call a platform for success. We focus on an ecosystem that promotes co-collaboration and has generated quite a bit of success. We've had over 6 IPOs and have created over $3 billion of company value. We believe strongly in this model, and we will be replicating it for a new project called B Labs at the Sierra Center, located in Philadelphia, with our partners Brandywine Realty Trust. We're currently doing tours for potential members, and we will be open by January 1st, 2022. If anyone's interested, please contact me directly. I'm also happy to announce that our brand new, much needed 37,000 square foot building with 15,000 square feet of lab space will be open in September of this year. It's been a long time coming, and it's much needed. We have a lot of key partnerships and events here at the Pennsylvania Biotechnology Center and Bloomberg Institute. I also would like to thank our parent nonprofit, the Hepatitis B Foundation, who is co-founder of this beautiful biotech center. We have 2 new school programs starting in the fall. A Master's in Biotechnology from Harrisburg University and an MBA in Pharma from St. Joseph's. I also want to make everyone aware of an upcoming Distinguished Speaker Series featuring Randall Heyer, the SVP of Moderna, taking place next week, July 29th at 11 a.m. This is a virtual event. It will be free. And for more information on the seminar, the new school programs, anything that I just mentioned or talked about, please go to bloomberginstitute.org. That's bloomberginstitute.org. So to learn a little bit more about the PABC, as we like to call it, in our community, we'd like to show you this video. We've been at full capacity for the last 8 years. There's been a tremendous demand. We've had a waiting list for most of that time period. When our new building opens up in September, it's going to double our lab space. It's going to be state-of-the-art, and it's going to help us fulfill some of that demand, although we will be at 100% occupancy when we open in September.
I always thought that the way we were going to help our cause for hepatitis B was to surround ourselves with innovators and entrepreneurs who were not necessarily working on hepatitis B or liver cancer, people with innovative ideas and therapeutic approaches that would make sense for us and would make sense to them.
This is actually my 3rd company that I started from scratch and my 3rd incubator space. But this has by far been the best incubator space that I've been in because of the collaboration, because of how open it is. I think that Tim Block is a genius. The way he's been able to create this environment with, with academic research, with his emphasis on translation, bringing the companies, and his openness to sort of share his experiences. And I think that pervades across the center, that, there's a culture here that, we like to interact with each other and learn from each other's experiences.
So it really is a two-way street for those companies that are more experienced, who have been around longer, and those new companies. We're always interested in more formal mentoring aspects to what we do. And of course, it's hard to say no if someone asks for help.
There are a lot of scientists who have good ideas. Most ideas fail, not because they are bad ideas, but because they don't find proper home, proper resources. This institute provides that kind of support. Moreover, scientists like me who came here were eligible to receive NIH grants.
The older companies, the senior companies can help out and give that advice to the younger companies. One of the most important things in this industry is how do you commercialize? How do you move your product further? You are going to hit road bumps. How do you react to them? And having people in our community that have been there before, know how to commercialize, who've done it, really helps out the younger companies. Excellent. And now it's time for the show. It gives me great pleasure to introduce our MC for today's event, Grant Castor, who is a close friend and advisor of the PABC, along with being a very successful entrepreneur. Grant is also the VP of Commercial Operations at Sentinel Therapeutics, and in his spare time, a firefighter. So please welcome Grant Castor. Thanks, Grant.
Hey, thanks, Lou. Appreciate the warm introduction. Pleasant good afternoon, everybody. This afternoon, we have 3 great presentations by entrepreneurs who are building diverse organizations based on incredible science. Before I introduce the panelists for today's event, I'd like to share a few fun market facts from the last year. So here are some highlights of 2020 in my view. Start of 2020, the pandemic hits the world and has a profound impact on our healthcare odyssey. According to a recent IQVIA data report that I read, there's currently a diagnostic gap of about 19% when you compare pre-COVID-19 diagnostic levels to 2020 and early 2021 diagnostic levels. So what impact does that have? Potentially later cancer diagnosis, later treatment on potential chronic diseases, less access to psychological and social services. Further, according to CDC, the US life expectancy fell to its lowest level in about 20 years, 77.3 years of age. They note a lot of the decrease can be contributed to COVID specifically. However, I would hypothesize that we will only see a minor increase to life expectancy over the next few years, simply due to the lack of this diagnosis over the last year. Further, the pandemic has caused a huge backlog in FDA field activities. So if you're an entrepreneur nearing a major milestone with the agency where it requires them to come visit your manufacturing facility or do a PV audit, etc., who knows when they're going to get to you. Currently, these backlogs have impacted about 48 new drug applications, and it's unknown what the full impact is going to be in the end. Further, about 1/3 of field activities by the agency occurs ex-US. So what does this mean for entrepreneurs in our space? Here are some of my predictions. I believe there's likely going to be an increase to capital in the coming few years. SPACs have raised an incredible amount of capital. Analysts expect that trend continues. However, the issue seems to be that SPACs are struggling to deploy capital once funded. As an entrepreneur, I see this as a huge opportunity. Further, public and government funding of early stage development for both diagnostic and therapeutic companies are expected to increase. I view this as an opportunity for non-dilutive funding for entrepreneurs out of the gates when they're the most exposed. Further, it's an observation that the market is becoming more niche. I believe there's a significant opportunity for individualized therapies. There's a heck of a lot less blockbuster drugs and therapies hitting the market. And further, at some point, I am optimistic that the diagnostic and therapeutic odyssey will intersect, truly transforming our system. So with that, it is my privilege to introduce you to our esteemed group of panelists for today's event. First, I'd like to introduce Mr. Eric Heel. He's managing partner with Medical Excellence Capital. Dr. Michelle Cleary, CEO of the Mark Foundation for Cancer Research, and Dr. Stephen Cohen, Executive Director with Daiichi Sankyo. We will kick things off with our first presentation, but before we begin, just a few quick reminders. We will have a 5-minute Q&A after each presentation. You can submit questions via the live Q&A button on your screen. Note, we will not have time to answer every question, and I highly suggest you attend the breakout sessions at the conclusion of our main session today. So with that, Eric, the virtual stage is yours.
Great, thanks, Grant. It's a pleasure to be here, and thanks for the invitation to participate. As you highlighted, we have all experienced in the past year with COVID, and despite the initial impact of the first generation of vaccines, we are far from done with COVID. And so it's my pleasure to introduce Dr. David Horn, the CEO and founder of Mid-Atlantic Biotherapeutics. David has over 30 years of research and clinical experience across infectious diseases, covering bacterial diseases, fungal diseases, and viral infections. David's experience led to senior positions at Bristol Myers Squibb, Merck, Thomas Jefferson, before founding Mid-Atlantic Biotherapeutics. David, I'll turn it to you.
Thank you, Eric. I'm going to share my screen now. Very good. Well, thank you, everyone. I'd like to thank everyone at the center for sponsoring this as well. So I'm here to talk about Mid-Atlantic Biotherapeutics. We're a company doing clinical stage immunotherapy plus vaccines. The company was founded about 10 years ago, and over $23 million has been invested in our IMT-504 technology. We also have a vaccine program addressing COVID-19, pan-coronavirus vaccines, as well as rabies vaccines. We have a therapy program, or a single immunotherapy, I should say an immunotherapy program, where we're using IMT-504 to address rabies, specifically rabies in the brain. And this is a fully partnered program that is fully funded. We are seeking $10 million to fund other key activities. IMT-504 is a patented, really very special sequence of DNA that supercharges the human immune system to fight infections. It's a mature asset. It's been in development for over 10 years. It's been used in multiple human clinical studies and has been studied as well in many different monkey models. It turns out to be very safe, with a strong safety profile. We only need to use very, very low doses of IMT-504. The advantage of that, it translates into a low cost of goods. It is also stable, easy to manufacture, and really can be scaled upward rapidly and economically. So what is the IMT-504 advantage? Well, basically, this special piece of DNA induces robust immune responses that allows our bodies to effectively and naturally eliminate viruses and also to make better vaccines. So what IMT-504 does, as you can see in the schematic, it really triggers both innate immune responses and adaptive immune responses in a very powerful but safe way. And what it does, it induces a lot of cells and messengers between these cells. So it leads to a very well orchestrated immune response. So we see macrophages and neutrophils that are important in cleaning up, scavenging, killing viruses, for example. We see B cells, which are very important in making antibodies. We see T cells are activated and proliferated by IMT-504 to induce very desirable killer functions. We have dendritic cells, which help orchestrate adaptive immune responses, natural killer cells, which you've heard a lot about in the cancer world, has killer functions to kill infected cells. And then all this orchestration goes on because of messengers called cytokines. And we induce a very highly desirable response. So when it comes to vaccines, the IMT-504 advantage is that it is a vaccine enhancer, also known as an adjuvant, and a key ingredient in developing next generation vaccines. It leads to a really marked boosting effect of the power of the vaccine, which leads to higher antibody titers, longer lasting vaccines with broader protection that require less vaccine injections. And very importantly, we see cross protection against viral variants. In immunotherapy, where IMT-504 is used by itself, it very effectively activates the immune system to fight people already infected, for example, with highly lethal viruses such as rabies. So this is really an excellent time to be in the vaccine marketplace. The vaccine market is projected alone this year to peak at $67 billion, and that's billion with a B. So as I mentioned, we're using IMT-504 as a vaccine enhancer or adjuvant to increase the efficacy of COVID-19 vaccines to protect against viral variants. And the key to all of this, because this is in the news every day, is to achieve very high neutralizing antibodies. The much higher is much better, because as we see these viral variants, it chips away at these antibody protection. So if you start at a very high level, you will maintain that level despite terrible viral variants being created. And not only that, we see an accelerated pathway to clinical development with a shortened time to launch. So why do we need better COVID-19 vaccines? Unfortunately, the current honeymoon for COVID-19 vaccines may be over very soon, sooner than we think. Multiple injections will be required to maintain protection over time. And the bottom line is the world, to get out of this mess, needs vaccines with greater durability, broader protection, specifically against the current and even future viral variants. So one of the questions that always comes up is, how do you compare one vaccine to another? The ideal way of doing it is a comparative clinical trial. That's the gold standard. But in reality, this is rarely performed due to cost, quickly changing landscape of vaccines. Neutralizing antibody titers may offer a way as a correlate of protection from symptomatic COVID-19. The higher, the better. So, for example, in a recent preprint, a vaccine efficacy of 80% against symptomatic COVID-19 was achieved with an antibody level of 247 in a live neutralization test. So here's our product profile, where we're comparing our COVID-19 vaccine with IMT-504 versus leading competitors. And as you can see at the top, we're achieving very high neutralization. These titers are very high. These antibody titers are extremely high, greater than 2,700 in our animal models. And the rest of the schematic compares our vaccine to the vaccines that are actually in humans. So you can see Moderna is achieving a titer of 654. Pfizer is hitting a titer of about 532. And the other two companies, AstraZeneca, J&J, have more modest results. So we're looking at neutralizing titers as a surrogate marker for vaccine efficacy. And scientific studies strongly suggest that higher titers lead to a more sustained and durable immune response. We're also working on a pan-coronavirus vaccine. There are other coronaviruses besides COVID-19 that cause disease. We have the original SARS-1 from 20 years ago. More recently, we have MERS. And by the way, 25% of the common cold is actually caused by common coronaviruses. So making a pan-coronavirus vaccine for now and for the future is actually really important. So we are using the unique properties of IMT-504 to fulfill that promise. And we are already partnering with two leading academic facilities to advance our vaccine. So on another note, I'd like to talk about rabies, because we've identified a rapid pathway to early FDA approval and revenue. So rabies, or rabies therapy in this case, is required, not so much in this country, but very drastically overseas. So typically we see a bite occurring from a rabid animal, but in the setting of no vaccine given, the virus ascends to the brain and the actual disease of rabies with impending death occurs. And there's no option at that point. Every person, 99.999% of people will die when rabies has reached the brain. It is the most lethal virus in the world. And again, there are probably over 70,000 deaths per year. So there is no current treatment or cure for rabies. And that's the big opportunity. We have partnered with a publicly traded company called Curative Biotech, which will fully fund our program. We would be the world's first and only treatment for rabies. A single human study would be required and could lead to a very rapid FDA approval. We have the potential for a priority review voucher sale, and these have gone up. These are currently at $100 million or greater. And in addition to that, we could start generating revenue within 2 years. So part of the rabies opportunity is also the orphan drug designation that we have received from the FDA for IMT-504 immunotherapy of symptomatic rabies. I mentioned the priority review voucher, and both of these things are very important FDA incentives for advancing drug development. This is our robust, diverse pipeline, and what we're seeing is at the top, rabies, the orphan designation, FDA approval anticipated 2023, the partner being Curative Biotech. The COVID-19 vaccines, we're seeing a preclinical program, but we own all the worldwide rights at this time. And the rabies vaccine, additional rabies vaccine program, is in the discovery phase. So we are seeking an investment of $10 million to fund key activities, including the completion of monkey COVID-19 challenge studies, the selection of the final vaccine candidate for human testing, and manufacturing scale up. And again, the rabies program is fully funded by partnership, so we're not seeking additional funds for this program. The value proposition includes the urgent need for a second generation COVID-19 vaccine, as I have laid out. There's an urgent need for a rabies therapy that actually works. And we see significant near-term return for investors. We're exploring IPO and a diverse reverse merger opportunities. So we are ready to go. We have great leadership, including myself. We have Dave Jobs, who has over 25 years of research and business development expertise at various locales like the NIH and other companies. We also have JP Gagnon, our SVP of Commercial Operations, who's had a very distinguished career at Merck for over 35 years, launching several multi-billion-dollar brands in the US. So I thank you for your attention.
Thank you, David, for that excellent presentation, and congrats on all the progress to date to get to this point with both your rabies treatment and then the enhanced vaccine program. Just a quick question to open up Q&A for the group. This pan-coronavirus vaccine enhancer, the adjuvant that you highlighted with IMT-504, do you see its efficacy being applicable to all of the current first-generation vaccines, or is it likely to work with one or more of the a specific type of first-generation vaccine?
So the reality is we have our own vaccines, which contains IMT-504. The reality is in the developed world, for sure, most people will be vaccinated. They will be vaccinated, if not now, relatively soon. And so the question is, what do they get next? How do they enhance their immunity with the next round of vaccines? And that's where we really fit in, because we believe our vaccines will enhance that immunity in anyone that's received any of the prior vaccines, and do it very notably as well.
Got it. Thank you.
Great. So friendly reminder, if you have any questions for Dr. Horn, feel free to submit them via the live Q&A button on your screen. So while we're waiting for a question or two, I'll ask a question. So you mentioned, PRV, or the priority reviewed voucher, and you also mentioned orphan drug exclusivity. What's your vantage point on Congress's extension of the PRVs to, I believe, September 24? So if for some reason you have a hiccup in your development timelines, how does that put your company at capital risk exposure past that timeline?
Well, I mean, we have our fully partnered program. It will be paid for. There will be revenues generated with success and other accolades because of that, and validation of our company and IMT 504 itself. So there's great value in that regard. Regarding PRVs, they've been around for a while. They're incredibly valuable. Speaking for myself, I would anticipate that this program will be extended. It has been successful. I believe the peak in value has been $350 million. The last sale was about $100 million in January, but the value seems to be rebounding upward again. So I think the PRV is a great, enhancement, and we have a great opportunity to potentially get one.
Great. So a question from the audience. This comes from Rahman Kapoor. What is the difference between the first versus second generation of the rabies vaccines? What are the shortcomings of the first vaccine that the second one would need to overcome?
So the first rabies vaccine, the way it's used is, first of all, they're extremely expensive. They cost a lot to make, and part of it are the ingredients. So if you can make a vaccine where you can dose spare, you're saving a lot of money. That has tremendous economic value. That's a property of our vaccines in general. The second point is that here in the United States or elsewhere, if you get bitten by a rabid dog, you go to the emergency room and you're obligated to get 4 vaccine shots. If you don't get all 4 of them, you very well could die from rabies. So the goal is to cut that in half, basically cut it down to 2 injections, or maybe potentially even 1, with a very effective rabies vaccine. So that's basically the hallmark, is that there's economic great value and other value in making a better rabies virus vaccine.
Got it. Thank you. And one last question before we turn to our next presenter. How difficult will it be to add excuse my butchering of this here, your adjuvant to a vaccine, particularly at scale?
So it's actually, that's a great question. It's actually very easy. So every formulation, every vaccine that we've studied or made, there's nothing fancy. You're taking your vaccine part, which is the antigen, the material, and just adding IMT 504 to it. So it's a very easy formulation from that perspective. There's nothing fancy or expensive in adding IMT 504 to a specific vaccine formulation.
Great. So I'll open it to our panel to see if they have any last questions before we move to our next presentation.
Maybe just, just a quick word of thanks. Thanks for sharing the story today here with us in the audience. Obviously, a timely conversation given the emerging variants and the need for second generation vaccines here. So thank you, David.
Thank you.
Thank you. All right. And with that, the virtual stage is yours, Dr. Cleary.
Thank you. I hope everybody can hear me. And it's a pleasure to be part of this event. And it's also a pleasure to introduce Richard Pestel, the president and CEO of Ecogenome. Dr. Pestel is an accomplished entrepreneur. He's founded 3 companies, one of which has had recently a highly successful exit. In addition to that, he's a world-renowned physician scientist who trained at the University of Melbourne in Australia, where he received a PhD in addition to an MD in medical oncology. As a postdoctoral fellow and a medical fellow at Harvard University and Massachusetts General Hospital, Dr. Pestel studied the biology of cell cycle control, and the results of his research substantially has shaped our understanding of this field and how it relates to cancer biology. Throughout his career, Dr. Prestel has held several senior level positions at Northwestern University's Feinberg School of Medicine, Albert Einstein Cancer Center, Georgetown University Medical Center, where he actually served as the director of the Lombardi Cancer Center. And more recently, Dr. Prestel has served as the director of the Sidney Kimmel Cancer Center in Philadelphia, Pennsylvania, and as executive vice president of Thomas Jefferson University. In 2018, CytoDyn actually acquired Dr. Pestel's company, Prostagene, for which he continued to serve as CMO and board chair until 2019. And currently, Dr. Prestel is a member of the Wistar Institute for Cancer, the Cancer Center in Philadelphia, and he's also Bloomberg Distinguished Professor. Today, Dr. Prestel is going to tell us about his company, Ecogenome. It's a company that's addressing a huge unmet need for cancer patients. They're creating theragnostics that will determine whether an individual patient will respond to a treatment or not based on tumor genetics, as well as the protein milieu of the normal cells in the tumor microenvironment. And this is going to lead to more optimal treatments that are tailored to the individual patient's tumor and will spare patients who wouldn't receive a benefit from a specific treatment, suffering those toxic consequences of some of the existing standard of care. So I'll turn it over to you, Dr. Prestel. We're looking forward to your presentation.
Michelle, thank you so much for that kind invitation, kind introduction. Can I just confirm you can see my screen? Perfect. Well, I wanted to start by thanking the organisers and in particular acknowledging the work that Tim Block and Luke Hasser have done to create a very unique scientific ecosystem in the Bloomberg Institute, which allows the Pennsylvania Biotechnology Center to flourish. You heard some of the numbers today from Lou about the effectiveness of this environment, but I can tell you as both an academic and as an executive leader, this is a very difficult task that they've achieved a superb outcome. It really is a very unique and successful environment. I moved my laboratory and my appointment to the Bloomberg Institute, as Michelle mentioned, several years ago with the intent of developing several biotechnology companies. And I'd like to speak with you today about Ecogenome. As Michelle mentioned, one of the key challenges in oncology, where I've been working now for almost 40 years, has been how to design a clinical trial in a way in which you optimize the population of patients who will benefit from that therapy. And secondly, what are the genetic predictors or other types of predictors that will determine what type of treatment will be optimal for the individual patient. And with those challenges in mind, we founded this biotech company in February of this year, although it's based on 20 years of research in this particular area. And we have brought together a very experienced management group and a laboratory team that knows this area extremely well. This is a forward-looking statement. I intend to give a quick overview of Ecogenome, intellectual property, some of the data underlying our conclusions, and the proposed financing. So I'd like to start with, at a personal level, with the notion that one of the most serious challenges we have today relates to the side effects of the therapy that we give to patients for cancer. Just to give you a practical example, about a third of children are going to die from the side effects of the therapy ultimately that they receive, particularly some subsets of children's cancers. And these side effects are serious and they're unwanted. And so part of our focus has been on how do we improve, by better understanding the cancer of that individual patient, the type of treatment they have, so that patients don't receive therapy that won't be of any advantage to them. Now, this is a schematic representation of a complex area which is part of the way in which many drugs that are used for cancer treatment work. And basically, there is an apparatus that is involved in repairing damaged DNA And there's this is a schematic of what's called non-homologous end joining. And there are many drugs that are currently being used which target this particular group of pathways which are activated by broken DNA. And I've listed some of these drugs on this slide just to give you a sense of the importance of this pathway as a therapeutic target. Now, this group of signaling molecules are in turn blocked by another type of pathway called homologous end joining. So this pathway inhibits the non-homologous end joining pathway. And this is a pathway which is targeted by a group of drugs called PARP inhibitors. And just to give you an example, this particular group of drugs has a market of around 4 billion this year. So this is a very large market. And one of the challenges in the PARP inhibitor field, but is true of all these different drugs, is how do we know which patients will benefit from the particular therapy? Now, as you can see, this whole process begins with the recruitment of a protein I've shown here called Q70, Q80. So this is the initiating event which activates all these different processes. And the fundamental question for all of us has been, how does this protein complex get chaperoned to the DNA? And we've identified a protein which plays a key role in this process called DACH1 or DUXUN. Now, the way in which we are focusing on solving some of these problems is to develop a test which can be used for the purpose of deciding the type of treatment the patient should receive. And the clinical trial design is informed in current practice by tests such as the HERCEP test. And clinician decision-making is informed by tests such as Oncotype DX. And this is an example of the HERCEP test, which is an immunohistochemical staining of a patient's breast cancer. And this is an example of the use of theranostics and Oncotype DX, which has been introduced into clinical practice. So in brief, Ecogenome is a spin-off from the Blumberg. We have strong evidence that this technology is able to predict with a high predictive capacity response to a variety of different types of therapies, and it's based on 20 years of development. This is the outline of the provisional application. So this gene was initially cloned in fruit fly as it blocks a hyperactive EGF receptor. We looked at its function and showed that it can block oncogenic signaling. It blocks the functional activity of cancer genes in a variety of different types of systems. And this is giving you an example of, this is a lung of a mouse, and these white dots are metastases. And when we overexpress DATCH, we're able to block those metastases. And so that led us to a series of investigations to try and quantitate the abundance of this DATCH protein. And this is a representative example of some of the early studies. DATCH is expressed in the eye, and this is the antibody staining in the back of the eye, the antibody we developed. And these are the early studies which showed that it is normally expressed in normal breast, but in different types of breast cancer, the abundance of the protein decreases as the tumor progresses. And we in turn have gone on to show that if you overexpress DATCH, you can reverse the formation of these, or growth of these breast cancer cells in a variety of different circumstances. This is the next sort of phase of academic clinical analyses shown here. This is an example of breast cancer studies, 2,115 patients, where a loss of DATCH expression substantially accelerated the rate of onset of the tumor and reduced survival. This is a subsequent analysis of 2,100 patients where we looked at the abundance of DATCH, and DATCH expression is lost primarily in triple-negative breast cancer. So these are 2,100 patients, the subtyping of their breast cancer. The abundance of DATCH is shown here. Red is increased and blue is decreased. We were able to illustrate that loss of the abundance of DATCH correlates with a substantial difference in outcome of these patients. We looked at lung cancer. These are examples of some of the lung cancer studies. Here we had 116 patients, and we showed that in lung cancer in particular, non-small cell lung cancer, there's a significant loss of DATCH expression, and this is related to the stage of the tumour's progression. And there's a substantial difference in prognosis in lung adenocarcinoma, again, over 200 patients. The most recent work we've been doing is in the area of prostate cancer. In this study of over 600 patients, again, DATCH deletion was associated with poor outcome prostate cancer, which really led us to this transition phase, which is looking at the commercialization of this test. But it's not the prognostic significance that led us to make this transition to commercialization, but rather the theranostic observation. Again, 320 patients, we find that the DATCH gene is deleted in up to 20% of patients, homozygously deleted, and heterozygous deletions up to 60% of patients. So this is not an uncommon finding in prostate cancer. This is a summary of 20 of the papers that published our work over the last 20 years, working and understanding the molecular mechanisms of DATCH. But the most recent observation, which is part of the patented finding, is shown on this slide. So let me share with you, this is a cell that has been irradiated with a laser. And when a laser radiates a cell, DNA damage occurs, and DATCH itself, which is linked to a green fluorescent protein, is recruited to that site of DNA damage, the damaged DNA, associated with the recruitment of Q70 and Q80. Those two proteins I told you were absolutely essential for activation of this non-homologous end joining. This is a quantitation of that data, the timing of the recruitment of those two essential proteins. But if we knock out the DATCH gene and make fibroblasts from those mice, laser radiation does not recruit Q70 or Q80. So DATCH is an essential chaperone, and this is shown schematically here, that upon the presence of double-stranded breaks, DATCH is recruited and in turn facilitates the recruitment of these Q proteins, Q70 and Q80, that in turn facilitate non-homologous end joining. Now, prediction of those studies would be that one might find resistance to PARP inhibitors, and it might enhance sensitivity to DNA PK inhibitors, and that is indeed the case. So these are representative examples of a large number of different types of studies that were done, in which we looked at the sensitivity of cells that were deleted of DATCH and their parental controls. And you can see that if we delete the DATCH gene with this particular DNA PK inhibitor, which is in clinical trials, cells are much more sensitive. If we reintroduce DATCH, we can reverse that phenotype. So DATCH is a key determinant of the sensitivity to DNA PK inhibitors. And conversely, PARP inhibitors, these are a series of different types of PARP inhibitors. This is a PARP inhibitor that's in development now. But in this case, you can see that the DATCH knockout cells are highly resistant to the PARP inhibitor. So this is a summary of the work that's been done to date. If we look in human prostate cancer cells that are deleted or knocked down of DATCH, or fibroblasts are deleted, there's a very high prediction of response, sensitivity to these particular drugs, and resistance, substantial resistance to these particular drugs. The work has been funded by a number of different grants from my research laboratory. As was mentioned earlier on, I have a background in executive leadership and in research in academic environments. Chief Scientific Officer Anthony Ashton has spent his life understanding how cells die and how drugs affect that. He was previously in Sydney as a scientific director of a large program there. The internationally acclaimed scientific advisory board include Elaine Mordecai, who has an exceptional talent at building biotechnology companies and spawning from them additional programs. In this case, his expertise in clinical diagnostics, particularly genetic-based clinical diagnostics, will be of tremendous value. And Chris Albanese, who has published widely in the area of molecular genetics, serves as the second scientific advisory board member. The research team, as I mentioned, has worked in this area for over 20 years. We have an experienced management team as listed here. And the use of these funds are to continue with in silico clinical trial database analysis and test the sensitivity of these drugs now in genetically modified mice. So we've developed mice which are selectively deleted of the DATCH gene to simulate what we see in patient samples. In this case, it's a prostate-specific knockout, but we can do it in other tissues. And this shows that we can delete the DATCH gene. We get a promotion, actually, of tumorigenesis in the prostate, and we'll be using these mice to test these therapeutics. Again, in summary, our intent is to lock down the assay in a clear lab and conduct technical validation of these studies with the intent of transitioning this technology to a strategic partnership, more likely than not as an LDT. So I'll stop at this point and answer any questions.
Thank you, Richard. That's very exciting science, and the potential for helping patients is also very exciting. I have a couple of questions for you. My first question is, the actual test in the real world setting, what would that look like? Would the physician have an app? Would there be a point of care diagnostic? Or would they be relying on the tumor sequencing that seems to happen more universally today when patients are diagnosed with their cancers?
Michelle, it's a great question. Our pathway will be to use immunohistochemistry, much like the Hercept test. A candidate would probably be DACO because they, of course, are the main, they are the main source of Hercept tests. And we would develop it with a strategic partner in mind. The reason for looking at the protein levels then, rather than just the RNA or the gene sequencing, is that we have found that in addition to gene deletion, which is frequent, as I mentioned, in those 4 large databases, a number of patients have a reduction in the abundance of DATCH due to methylation of the promoter region of DATCH, which leads to silencing and reduced expression of DATCH. But your point is well taken, which is it's a relatively simple series of steps now to take the work that's been done in the academic environment and transition it to a clear lab based on the really substantial amount of experience we have in the academic environment. There are a number of other laboratories that have subsequently worked in this area. And I should mention that there are a number of other cancers beyond those which I've shown you today. And so that in addition to lung cancer, adenocarcinoma of the lung, breast cancer and prostate cancer, in glioblastoma multiforme, which is a very deadly form of brain cancer, the large Japanese cohort has shown a substantial reduction in the abundance of DATCH in those patients due to gene deletion. So there are actually a large number of different cancers in which DATCH is either deleted or methylated and silenced, so we need to look at the protein abundance.
My second question for you is, Richard, you highlighted that you were able to get to this point with over $10 million in government funding. What would your advice be to another rising entrepreneur who wants to get the early stages of work done through non-dilutive type funding before taking something into the commercial realm?
Well, Michelle, it's a great question. You know, my impression is that we have learnt a great deal over the last 10 years about, or almost 20 years about this protein and its function. And it's only very recently where we discovered the theranostic significance. And I think it's about identifying the point at which you need to transition into the non-government funded transition. In the case of Prostagene, we wanted to conduct a clinical trial, and so it was important to have a publicly traded company as a vehicle for raising funds on Wall Street, and had the opportunity to raise about $50 million, which in turn led to a multi-center national clinical trial on triple-negative breast cancer. And that could not have been done through, to the best of my knowledge, through the NIH processes, and certainly not as quickly as it occurred in this case. So again, in answer to your question, when to take government funding versus investor funding, I think it really depends upon the moment of catalyst of value creation.
Thank you.
Great. So reminder, the live Q&A is available. I'll open the floor to our panel real quick to see if they have any questions. All right, I'll ask just a pointed question. I know you summarized or you at least put the funds you're looking to raise in the short term here. So what is the total amount you're looking to raise, and are you looking for one investor or are you looking for numerous checks in that round?
Well, I think the amount we're looking for is 1.5 to 1.7 million, and the process by which that comes, whether it's one or multiple, I should add the caveat that the prior investors in Prostogen had expressed an interest in co-investing PABC in any future ventures that I'm involved with. So it would be to, we would welcome either small numbers or a single investor, but there's a caveat that the prior investors in Prostogen wanted the opportunity to co-invest PABC with the new investors in this company.
Great. Last question from the audience here, and then we'll move to our next presentation. What exactly is the patented intellectual property if the research is in the public domain?
Yes, the information that's not in the public domain, which hasn't been published, which is in the patent submission, relates to the role of DATCH in DNA damage repair, which I shared with you today, and the prediction of response to therapy. So the theranostic information, which I shared with you today, is not in the public domain. It was the basis of the patents. So this transition, as shown here, is the in the green box is the DATCH determines therapy response and the mechanism of action thereby.
Got it. Yeah, Dr. Pestel, your screen is not on the main stage right now, so possibly something to cover during the breakout session.
Can you repeat what you just said? You just
broke. So your screen is not currently on the main stage. So perhaps we could cover this in the breakout session following the main session here.
You got it.
Great. All right. So with that, I'd like to welcome our next panelist, Dr. Stephen Cohen, to introduce our next presentation. Dr. Cohen, the virtual stage is yours.
Yes, thank you very much. It's an honor to be here today, and thank you very much for choosing me as a panelist. I also, with Lou's permission, wanted to mention that Daiichi Sankyo's association with PABC has been enlightening. We're exposed to cutting edge new science that hopefully will eventually be translatable into drugs that we can use to meet unmet medical needs. And to that end, it's my honor to introduce Dr. Minghang Zhang, who's an accomplished scientist, received his PhD in chemistry from Brigham Young University, his postdoc from Yale in biochemistry and biophysics, and completed a fellowship at the NIH before beginning his trek into pharma for the past 14 years, most recently as principal scientist at Janssen, part of J&J. 2 months ago, he decided to leave Janssen to join Gene Lancet as their CSO, a company which began in 2019 with one of his patents. And with that, Dr. Zhang, the stage is yours.
Dr. Steve Cohen. First, I want to say, I need to thank the organizer, PABC. We are new members, so. And Gene Lancet is a biotech company. We develop a star editor, and it is a CRISPR-based gene editor for Scarlet cure. CRISPR editor is an investigational drug, and it has a great potential to cure many diseases. And today, I will take this opportunity to introduce you to our chronic HBV program. Chronic HBV is a persistent worldwide health problem. And in many countries, there are over 1/10 population, get infected by HPV, as shown in the map in deep blue. So, and their annual deaths around there's 0.6 million worldwide. Sorry. And the current standard of care for chronic HBV include nucleoside analogs and interferons, and also there are other chemical modalities in development, such as siRNA. And most of these existing, treatment regimes and developing modalities have a universal issue of viral rebound. That means when the patients get treated and this, with these drugs, and this is a lifelong treatment. And since this is lifelong, and it has highly possible to develop a drug resistance, that means, the drug could, finally does not work. And many patients actually go through that liver cirrhosis, and this leading to that HCC. And some other patients may not go through liver cirrhosis, but the chance of have that HCC is much higher. And currently, this is a major issue, as I mentioned, and surely, many companies, including us, looking for cures. Particularly, the scientists in the field are looking for functional cures. It means, to get rid of that HBsAg, the surface antigen. Unfortunately, as I mentioned, this is a mission impossible based on these current existing drugs and also those drugs in development. It has already been discovered, the direct cause of chronic HBV is that cccDNA. Shown below is a simplified, picture showing that the life of HBV and hepatocytes. And the HBV DNA usually exists in two forms: that the relaxed circular DNA and also double-stranded linear DNA. Both of them form cccDNA, and the double-stranded linear DNA actually also give rise to, the DNA integrated into human gene. And that integrated human, that integrated HBV DNA can actually form a lot of, the viral antigens, including HBsAg, which, known to, leads to inflammation and also other, many complexities. So here, one thing is sure: to cure HBV, we need to, stop expression of proteins or replications of all forms of viral DNA, and gene action targeting, viral DNAs provide an opportunity, to cure this disease. Unfortunately, therapeutic CRISPR is still at its infancy. It has many collateral damage, which could kill patients and even affect future generations. So the CRISPR-Cas9 is actually a binary system. It is composed of guide RNA and also the protein, the enzyme. The guide RNA is a single guide, RNA. It directs where the enzyme cuts, and then it's up to the cell to repair the cutting, to fix whatever this cutting may result in, and then for that correct accurate repair, the cell requires a template. This template is usually introduced by co-delivery with the enzyme, and so far, for the in vivo gene editing, the efficiency is extremely low. So, and in addition, single guide RNA is really long and is well known for its complexity in manufacturing. Lancet developed a star editor. We developed a star editor with a minimum collateral damage for sclerosis cure, and star editor comprises our internationally distinct guide RNA. Is still single molecular guide RNA, but is LgRNA is comprised of our intellectually distinct non-nucleotide linker, and this LgRNA can be produced with high quality. Thus, it will minimize off targets and also other collateral damage as mentioned. In addition, it includes a single-strand DNA for accurate repair. For our chronic HBV program, the star editor actually comprises a specially designed LgRNA, and it deactivates all HBV DNAs. So in that way, and this star editor may get rid of most of HBV DNA, and then surely your immune system may finally just kick it in, get rid of most of them, leading to a complete cure. For our delivery, right now we are still using, thinking of that nanoparticle LNP, and but we also develop that CRISPR conjugates for liver delivery, but also targeting other organs. And star editor has already been proven to work, more efficiently than the other wild type CRISPR system, as I showed here on the right side of the slide. The 1 to 4 lengths actually are shown that the star editor, including LgRNA, IVT is a wild type of CRISPR Cas9, and there are 3 rows, and the top 2 rows, the top rows is actually uncut product, and the bottom 2 rows are the product. And those numbers are the efficiency of editing. As you see, star editor has a better efficiency. So I want to point out our chronic HBV program itself is highly profitable, simply because this is a huge what health problem. So as you see, there are over 257 million patients right now get infected by this chronic HBV, and 2/3 of them have liver cirrhosis, and actually around over 0.6 million of annual deaths due to this complexity of this later stage liver cirrhosis and HCC. And what's more, the patient pool is still increasing. And as you see, annually there are still 4.5 million new infections. That means, the burden for this water health system is actually, still a huge issue, increasing yearly. And but I want to also point out, star editor targeting pathogenic DNA, and these can be used to cure many diseases, including HIV, Alzheimer's disease, cystic fibrosis, and also other monogenic disease. And as you know, most of the disease actually, the patients don't have any treatments simply because we couldn't identify effective drug targets, right? And and also we believe, there's a huge opportunity for anti-aging and also regenerative medicine, and so our current pipeline includes, as mentioned, is mainly focused on chronic HBV and also the platform enhancement. We are asking for $2 million for the seed round to complete proof of concept in animal models, and also advance our leader program to the GLP tox readiness. And we hope at the beginning of year 24 to start a clinical trial. And at that time, we also hope to expand our pipeline to HIV, HSV, and also other monogenic disease. And we hope, the company is scheduled to, start our anti-aging and regenerative medicine after the company's IPO. I just want to point out that Gene Lancet, has brought over a really prominent investment opportunity. As I showed here, we actually have products, the LGRNA related products ready for commercialization, and also for our, the star editor technology, we definitely can get the additional revenue stream from, strategic collaboration with other pharmaceutical companies, and also based on the technology licensing. But I do agree, for that the chronic IGBV program may need quite some, the fund infusion. However, we are really confident in year 3. We think that our EBITDA should be positive. Sorry, I cannot see anybody. I just, but I want to point out Gene Lancet is a young company at a really early stage. And so far we have secured the exclusive US market with our LGRNA related products. And our star editor technology is worldwide patent pending. And we are still building our team. And the current team includes the 2 founders, me, and and also 1 collaborator. And as Steve mentioned, I'm an experienced scientist. I'm working in industry for 14 years, particularly in infectious disease. And Sherry is an experienced program manager. She has her own company before too. And Dr. Guo is actually a top HBV virologist. He's at the center. I think, this is my last slide. At the Gene Lancet, we always put the patients first. And please, help the patient by supporting us. Thank you. I'm ready for questions.
So maybe Minghang, I'll start with the first question. First of all, thank you very much. This is exactly what I was referring to when I was saying that PABC represents a lot of the cutting edge new science, and why a lot of people want to be associated with the PABC. My first question is really about maybe your competitors. The gene editing had been on the cusp of the new cutting edge science. There seems to be papers about this almost every day that I see in the literature. How would you differentiate your platform from the many others that seem to be popping up over the past few months and years?
Thanks for this nice question. And yeah, absolutely right. This is a hot zone. I have to agree. So actually, there are many companies. At least people can think of the 4 giants right now, right? And CRISPR Therapeutics, Engineer, Editas, etc. However, for most of them, as I mentioned, for CRISPR-Cas9, whatever generation of gene editor you have, you still need that single guide RNA. The single guide RNA, as I said, directs where the enzyme cuts. And unfortunately, single guide RNA is 100 nucleotide long. It's really complicated to synthesize to get it pure. And for us, we have that cutting edge technology of LGRNA. We can provide high quality RNA. And what's more, in our LGRNA, we included a ssDNA template. That means it can direct the accurate cellular repair. And for other systems, most of current compounds in clinical trial are ex vivo, and those are lockouts. So surely, for in vivo therapy, that's not even related. And surely most of the recent news should be about the engineer still is a lockout strategy. And for us, we hope to develop in vivo. We hope our star editor will lead the age of this area, and our LGRNA technology can enhance the platform, not just for us, but for the whole field. We think that our intellectually distinct technology will actually contribute to the field itself. Thank you, Steve.
Thank you very much. Grant, are there other questions coming up?
Yeah, so a reminder, the live Q&A is open. While we wait for some questions, I'll open it up to our panel if there's any additional questions.
I have a question. This is really beautiful that you can package these into nanoparticles, and being that it's nucleic acid, it's probably also amenable to some of the other newly burgeoning delivery strategies like modifications. Are you exploring any of that so that you can get wider tissue distribution than just liver?
Yeah, that's a good question. Actually, delivery for the gene editing, almost any system, is really challenging right now. Although, engineer recently reported the data, they can get really, really great, data for the liver delivery, right? And, for us, surely, LNP is our first choice because we target the liver for the chronic HBV. But at the same time, as I showed a little bit on that slide, we actually develop conjugates of CRISPR case. That means we will have those, the IMP modified with ligands or even antibodies, for that tissue targeting. Those are in progress.
Great.
Thank you.
Great. One question from the audience. What is IgRNA?
Let me explain a little bit here. And as you say, most of the recent Nobel Prize was awarded for that CRISPR-Cas9 system, right? That's actually to point out the key point. That is sgRNA. That means they put that cRNA and the trace cRNA together with a tetra loop, that GAA, right? So then you get a single piece of RNA. You can do that in vitro transcription, and you can also deliver that as DNA. Unfortunately, we believe for the future, particularly for the drug, for the medicine in human, maybe you all have to use a synthetic guide RNA. That means synthetic sgRNA. That is really long. For us, we developed that strategy. This is a patented strategy, by the way. And we actually put intellectually distinct long nucleotide link between that cRNA and the trace RNA. So that means for our synthetic, single molecule guide RNA is still a single molecule, but we can synthesize stepwise. We can synthesize the cRNA, actually shorten the cRNA, and also shorten the trace RNA, and then use a chemical ligation, and then to form a single molecular RNA. And what's more, by this chemical ligation strategy, we can also introduce single-stranded DNA as a template. And when we are talking about the LGRNA, that means chemically ligated guide RNA. And it could include a DNA template, but for many other lockout experiments systems, you don't need a single-stranded DNA. Then just that the chemical ligated 3 pieces of guide RNA. I want to point out that in our system, we even further divided the trace RNA into additional 2 fragments of similar sizes. So the synthetic efficiency is really high. The quality is much better. That's LGRNA and our LGRNA technology. Hope that answered your question.
Great. Well, amazing technology and science. So really appreciate the thorough answer. So with that, I'm going to invite Lou back up to the virtual stage. A really big thank you to our panelists and presenters. So with that, Lou, the virtual stage is yours.
Thanks, Grant. And I just want to remember in a few minutes, we're going to have Grant back on to talk about breakout rooms. So for those attending right now, you will have the opportunity to go into a room with each one of the three companies. You can pick which one you want to talk to as we wind down the program here. I want to take this minute, though, just as a reminder that the Pennsylvania Biotechnology Center is a nonprofit. Donations we receive go towards much needed scientific equipment, which everybody I'm sure on this event knows can be very expensive. And we also, any donations go towards our academic programs. Specifically, we have a high school program here that we host and run with the Central Bucks School District, where the students come here every day during the school year to take advanced chemistry. And then they get a mentor within our labs, and they go out and they do research. It's one of the best experiential learning programs in the country. The issue is the lab supplies for the students can be very, very expensive, and your donations will really help provide support for the scientists of tomorrow. So if you do have some time, I'm going to give you a phone number right now that you could text the word donate to 267-459-4990. Once again, text the word donate to 267-459-4990. Every dollar helps. It really, with our programming, with equipment, with academics, with students, we make it go a long way, and we really appreciate your support. The next item I'd like to talk about is our PABC membership. So it's some incubators have two types of memberships, and we do here. One is, of course, people who rent lab and office space are part of our community, but we also have close to 90 members, people who pay $125 a month, which is $1,500 a year, to be part of our community. What does that entail? The slide can give you some bullet points, but really it's all access to the Pennsylvania Biotechnology Center, our events, our seminars, our networking, different types of activities that we have, lunch and learns. You also will get a mailbox if you're looking to write a grant and you need a location, and we can help out with that. It's something that's a lot of value. And for today and today only, we are doing a $50 raffle. So a $1,500 membership, if somebody right now in this event goes to our PABC, let me get the right wording here, the PABCevents.org. Once again, PABCevents.org. You can purchase a raffle ticket for $50 and have an opportunity for this tremendous membership where you will be on our email list. You'll know what's going on, company news, and we like to have a lot of services and activities almost daily. Once we can open back up to where we were before the pandemic, you'll have every opportunity to be part of our community. So once again, it's PABCevents.org. And hopefully, and I'll talk to our events team, we can keep that open for the rest of today and maybe do the raffle tomorrow just to give everyone an opportunity, PABCevents.org. The next slide is probably one of the most important ones that we're going to show today. And it was a great event today, and the three companies did a great job along with our panel and our MC Grant. I really want to thank everybody. But we have to thank our sponsors. Without their support, this event would not happen, and a lot of the things we do here would not happen. So our sponsors are very near and dear to our hearts, and thank you so much for contributing and helping out with this event and other events that we hold. So at this time, before I pass it back to Grant, I want to just take a second. There were some technical glitches today, and I do want to apologize for that. I just heard, and once again, we don't know how widespread it was, but especially logging on, and some people would get logged off. And then with the video that we showed at the beginning, we heard that there may have been a part of it that wasn't completely shown. So before we go to Grant, who's going to give the breakout information so that you can talk to these companies one-on-one, we are going to play the video again. It's a very short video, but it just gives a brief overview of what the PABC is about. Thank you. We've been at full capacity for the last 8 years. There's been a tremendous demand. We've had a waiting list for most of that time period. When our new building opens up in September, it's going to double our lab space. It's going to be state of the art, and it's going to help us fulfill some of that demand, although we will be at 100% occupancy when we open in September.
I always thought that the way we were going to help our cause for hepatitis B was to surround ourselves with innovators and entrepreneurs who were not necessarily working on hepatitis B or liver cancer, people with innovative ideas and therapeutic approaches that would make sense for us and would make sense to them.
This is actually my third company that I started from scratch and my third incubator space. But this has by far been the best incubator space that I've been in because of the collaboration, because of how open it is. I think that Tim Block is a genius. The way he's been able to create this environment with, with academic research, with his emphasis on translation, bringing the companies, and his openness to sort of share his experiences. And I think that pervades across the center, that, there's a culture here that, we like to interact with each other and learn from each other's experiences.
So it really is a two-way street for those companies that are more experienced, who have been around longer, and those new companies. We're always interested in more formal mentoring aspects to what we do, and of course it's hard to say no if someone asks for help.
There are a lot of scientists who have good ideas. Most ideas fail, not because they are bad ideas, but because they don't find proper home, proper resources. This institute provides that kind of support. Moreover, scientists like me who came here were eligible to receive NIH grants.
The older companies, the senior companies, can help out and give that advice to the younger companies. One of the most important things in this industry is how do you commercialize? How do you move your product further? You are going to hit road bumps. How do you react to them? And having people in our community that have been there before, know how to commercialize, who've done it, really helps out the younger companies.
All right. Thank you, everybody, for your attendance today. Just a housekeeping item before we break out into the breakout sessions. So to navigate to the breakout sessions, simply go to the timeline from the main session. On the main page, you'll see the VI table of visit sections. Simply click join and wait for the breakout rooms to open. 3 blue buttons will appear on the screen, and then you will choose which company you'd like to join in the breakout rooms. Just in closing, I want to thank everybody for their attendance, our esteemed panelists, our amazing scientists and entrepreneurs. Look, it truly takes a village, and the PA Biotech Center is an amazing village. I'm very fortunate to have 1.5 miles from my home, and it's something that Lou, the team, Dr. Block, and everybody should be very proud for all their hard work and dedication they've put into building it. So again, thanks for your attendance today, and please navigate back to the breakout sessions on the timeline.